Hi, I'm Nathan Smith, this is Michael Lane, and our third person, Victoria Swiler, was unable to make it today. Our facilitator was Nicholas Whitaker, and we're from Athens, Ohio, and we're representing Ohio University. And so what we know about our understanding of science is based on the Earth environment. And gravity plays a large role in that. However, in microgravity, without the extent of gravity pressures, we can uncover and unveil pathways and mechanisms in plants and animals that we can't on Earth. And so what we tried to look at was the problem with sustainable agriculture on the International Space Station. Now, for long-term missions, you're going to need regenerative agriculture to sustain astronauts and produce fresh crops. As we saw with the astronaut talk, they have a lot of not fresh food. They have a lot of freeze-dried food. And so our solution to this, our possible solution, was plant growth-promoting bacteria. And plant growth-promoting bacteria, in association with plants, they can increase crop yield, they can fix nitrogen in a usable form for these plants, and they can produce phytohormones such as auxin, which is the main hormone in plant growth.
All right, so for our experiment, we wanted to investigate plant microbe interactions in microgravity. So we started with our bacteria, which is Sphingomonas sanguinis. We found a strain that was actually isolated from the International Space Station, so it's already adapted to microgravity. And the other reason we chose it was because Sphingomonas sanguinis is known to have many of the plant growth promoting factors that Nathan was talking about. And the plant that we plan to test it with is Arabidopsis thaliana, which is just a model organism that is often used in experiments such as this. And our hypothesis is that Arabidopsis and S. sanguinis will both have a mutualistic relationship where both will have benefited gene expression and increased growth. We plan to test it using our mini lab design, which essentially is going to have a divider straight down the middle, half of it with the Arabidopsis seeds grown in media, and the other half with Arabidopsis seeds in media with bacteria also in it. And then on the sides, we have RNA later, so that when the plants have grown an appropriate amount, the astronauts will release the clamps, and then the RNA later will bathe both the bacteria and the plant and arrest it the way it is, so that when it gets back to Earth, we can perform gene analysis and measure the growth the same way that they were while they were growing in space.
And so we do have preliminary data on this. And as you can tell, the picture on the right of your screen, far right, and that is an Arabidopsis thaliana plant not inoculated with bacteria. However, the picture beside it is an Arabidopsis thaliana plant with bacteria in the MS. And as you can tell, there's a great difference in the root structure, as there are many more root hairs grown with the bacteria. And this is important because root hairs are able to soak up those nutrients and water, and so increased abundance of those would allow the plant to possibly uptake more nutrients and be able to grow larger in space.
And then our other graph demonstrates the difference in the average mass between the plant material that was inoculated with bacteria and the plant material without bacteria. So essentially what we did was we cut it in half, so we had the root and the shoot of the plant, and we measured both of them independently. You can see that the blue part of the graph is the plant material that had bacteria on it, and it is much larger than the one without, which is very promising data, and we hope that will continue for our final experiment. So our research is very important for reasons beyond just creating sustainable agriculture in space. Plant growth promoting factors can also be, plant growth promoting bacteria can also be an alternative to pesticides, and that could be used here on Earth, which is a safer way to do it. It's also known to be a great biofertilizer. And then, of course, this experiment will expand research in the field of plant biology. And we would like to thank our sponsors and funding for giving us this opportunity. First off, I'd like to thank Ohio University. There's a picture of everybody involved with the SSEP right there. That is just the finalists, though. We had much more people. It was a great project. We'd also like to thank SSEP for giving us this opportunity to explore what we want to explore. We'd like to thank the NASA Jet Propulsion Lab and the California Institute of Technology for helping obtain some Sphingomonas sanguinis bacteria, because as you can imagine, it's difficult to get that from the ISS. And then we'd like to thank the National Center for Earth and Space Science Education for making all of this possible. Thank you. Are there any questions?
I actually have a question. The Step 2 National Review Board is always impressed by seeing new creative ways to use the mini laboratory. So in your particular case, you're using a type 3 with a divider, so you have two separate experiments going at the same time. So how did you manufacture the divider and make sure that there's no fluid moving between the two chambers?
Yeah, so the question was, how do we get the divider, and how do we know there's no cross-contamination between the experiments? Well, so that divider is actually a polypropylene piece of plastic. It is just the top of an Eppendorf tube cut off, and then we trimmed it around so it would fit in the tube. And then we can swab both sides of the mini lab, and then we can try to grow bacteria. And obviously, we want no bacteria on the control side, and we do want bacteria on the bacteria side.
Any more questions? Okay, thank you so much.